everyone! I'd like to show you an actual electrophoresis machine and its components. If you're planning to take more biotech classes at Ohlone, you will get an opportunity to use this as well as create a gel slab in one of the classes. Also, a huge thank you to Dr. Lori for allowing me to borrow a gel electrophoresis machine and its components to show you all. So first things first, this is the buffer chamber. This is where you put the buffer in as well as the gel slab. The buffer is a clear liquid that is used to provide a stable pH and a conduction of charge. When pouring buffer in, you first want to make sure that the gel is placed before pouring. This helps reduce the chances of buffer splashing out of the chamber or overflowing the chamber. You want to add enough buffer to cover the slab, at least 1 millimeter above its surface. On this chamber, it is marked for you, which is really convenient. These are the electrical leads. The anode, which is the positive lead, and the cathode, which is the negative lead. These two connect to a power source which will provide power to the chamber. The power creates an electrical current through the chamber for the samples to move through on the gel. You'll know it is on when you see tiny bubbles coming up from the ends here. If you're analyzing DNA, one thing you should know is that DNA is negatively charged, so this means that it will travel toward the positive lead. These two items are the combs, and when creating a gel slab, these are used to create the wells to pour in your samples on the gel slab. There are two different sizes, a large comb that creates 6 wells, and a small comb that creates 10 wells. This is the gel tray. This is where you place the gel slab on the machine. You can also use the gel tray to create your gel slab. You'd first need to seal these two areas with tape or with shields that your instructor may have. Place the combs in the designated area and pour in the agarose solution. An important note when making a slab is to make sure that the tray is on an even surface so that your gel slab does not contain uneven areas, thus creating distorted bands. This is the gel slab. It is made with agarose and buffer solution. When making the slab, you would have to combine both agarose and buffer solution in a large flask and heating them up and gently swirling the flask until the agarose solution is clear. You would have to be very careful when it comes to making the solution because it will get very hot and there are instances where it will bubble over in the flask. So please exercise caution. I'd also like to mention that in class, when your solution is slightly cooled, your instructor will add ethidium bromide to your solution. This will help see the bands on the slab much better under UV light. However, ethidium bromide is a mutagen and is toxic, so you have to wear gloves when handling your slab. The one that I am holding is actually a fake slab, so I don't need to wear gloves. Now, when placing your gel slab in the machine, the position is very important. As I mentioned earlier, DNA is negatively charged, so it will be traveling towards the positive. This means that the wells would have to be placed where the cathode, or the black negative lead, is located, just like so. That way, when the machine is turned on, the bands will be moving toward that direction. So let's focus a bit more on the slab. As you can see, these are the wells that were created from the combs. The lines that you see are called the bands, and these bands contain the DNA. You may notice that there are bands all over the slab, some closer to the wells and others that are closer to the other end of the slab. These indicate the sizes or base pairs of the DNA. An important thing you should know when it comes to reading a gel slab is that longer strands of DNA move slower through the slab, while short strands move quicker. So you may be wondering, how is it that we are able to see these bands? When pipetting our samples into the wells, we also add a staining solution called bromophenol blue. This helps to distinguish each band on the ladder. So from looking at this slab, you can tell that here are where the longer strands are, and here are where the shorter strands are. Another thing that I would like to mention when reading a slab is the importance of a DNA ladder that contains known sizes of DNA base pairs to help you determine the base pair sizes of unknown DNA that you may have in your sample. Normally, we would put a DNA ladder on the first or last well of a slab to not confuse ourselves with our other samples in the other wells. There is more information about DNA ladders on both videos that I have provided from YouTube. That is my video, and I hope you enjoyed it.